Assalamualaikum to those who have just joined. And today, inshallah, we'll begin with our second chapter. Technically, this is our third chapter, but as per the CAI, this is your second chapter, biological molecules. ठीक है? So, before starting off, किसी को पता है कि what exactly biological molecules are?
Carbon, hydrogen, oxygen.
Carbon, hydrogen, oxygen. But aren't they the elements that make up the biological molecules? Molecules, though, it's supposed to be big, while carbon, hydrogen, and oxygen, they're elements that make up those biological molecules. So technically, you're right, but in actual, biological molecules are carbohydrates, they're proteins, they are lipids. So elements do make up biological molecules, and mainly carbon, hydrogen, and oxygen are responsible to make biological molecules such as carbohydrates, lipids, and proteins. Okay? So let's begin, inshallah. I just mentioned that these are the biological molecules, and we'll be covering each one of them in detail. Number one, carbohydrates. Number two, proteins. Number three, lipids. Number four, nucleic acids. So in this chapter, we are not going to delve deeply into nucleic acids because we have a separate chapter of nucleic acids. I believe that's chapter 5. And when we'll begin with chapter 5, we'll cover nucleic acids deeply. But right now, we are going to cover carbohydrates, proteins, lipids, and water. Okay? So these are the part of these are the part of your syllabus, and we have to do them. And before understanding and before diving extensively into each one of them, we need to understand few terminologies. And I have mentioned all those terminologies over here in the first slide. So the first keyword that you need to remember is monomer. Monomer literally means that they are the simple molecules and they're responsible to make a large polymer. So you can consider them as the brick. And when we combine lots of bricks, they make up a building or they make up a wall, right? So a wall can be considered as the polymer, while a brick can be considered as the monomer. So monomers are the simple blocks that join to form polymers. And then polymer, so it's a giant molecule made up of repeating similar monomers. So this is a very important word that you have to remember, that monomers should be similar if we are making up a polymer. इसकी एक important reasoning is कि I'll let you know as we progress. So as for now, try to remember that when we construct polymers, they should be made from similar monomers. We do not have to mix up two different monomers in order to make a polymer. A polymer is always made up of same monomers. Now, moving towards macromolecule, macro means large, and molecules, obviously it means biological molecules. So large biological molecules such as lipids and proteins. Okay? So for now, just remember the fact that lipids are not polymers. Okay? Lipids are not polymers, rather lipids are macromolecules. And there's a reasoning that why I'm calling lipids as macromolecules and why I'm not calling lipids as polymer. Okay? And then, like Minha mentioned, that we have these important elements that make up these biological molecules. So different proportion of carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus are responsible to make different sort of biological molecules, but all of them like comprise of these basic elements. Okay? Then this is an important process, condensation and hydrolysis. We'll see deeply how different biological molecules are condensed and how different biological molecules are hydrolyzed. So when we have to make something, or when we want to synthesize a molecule, then we go through the process of condensation, where we have to remove the water molecule in order to construct some sort of biological molecule. And when we have to break some sort of biological molecule, we make it go under the process of hydrolysis. In hydrolysis, you have to add water in order to break a molecule, while in condensation, we remove a water in order to synthesize a biological molecule. So far, are we good? Have all of us understood whatever I have mentioned so far? Being confused to नहीं हो गए? All good? All right. Okay. So again, this is an important thing that you have to remember, that monomer should be made up of same no, sorry. Polymers should be made up of same monomers. Up here, let's see. So this monomer amino acids, when lots of amino acids they combine, they form a protein. Okay? So when various amino acids combine, they'll form protein. Similarly, when various glucose will combine, it will form a cellulose. Okay? And when a nucleotide, we are not going to study the structure of nucleotide in this chapter. We'll do nucleotide in chapter 5. But as for now, just understand the fact that DNA and RNA, the nucleic acids, they are made up of the monomer nucleotide. Just observe one thing, that I have not mentioned lipids in polymers, and I have not mentioned the monomers of lipids as well. And why is that so? Because lipids are made up of two different molecules, that are glycerol and fatty acid. So like I said, that polymers are always made up of same sort of monomers, right? So lipids have two different molecules, glycerol and fatty acid. This is exactly why we don't consider lipid as polymer, rather we consider lipid as a macromolecule. Reasoning samajh aayi? So far, are you good?
Yes, miss.
All right, perfect. So it's also
Miss, polymer और macromolecules में क्या फर्क होगा?
yeah, I'm explain it to you. Just wait for a few minutes. Acha. So now see that now you're not going to say that fatty acid and glycerol are monomers of fats or lipids, because यहाँ पे lipids are not known as polymers and fatty acid and glycerol are not known as monomers. Rather, they are considered as building blocks of lipids, and lipid is considered to be a macromolecule. So the difference between macromolecule and polymer is that polymers are always going to be made up of same monomers. Like they'll have same building blocks. But a macromolecule can be made up of different building blocks like lipids. Okay? So lipids में there are two different building blocks, fatty acid and glycerol. इसी वजह से we are saying that macromolecule is a large molecule and it has different building blocks. So lipid is not a polymer as it is made up of different monomers. Okay? So this is the reasoning. And you'll have to remember this. Never write lipid as a polymer, because lipid is not considered to be a polymer. Why is that so? Because it's made up of two different building blocks, which are fatty acid and glycerol. So far, are we good?
Yes, miss.
All right. All right. So now let's move on. Now we are going to study lipids. Then we'll study proteins, and then we'll proceed towards carbohydrates. So starting off with lipids, we just mentioned that lipids are made up of fatty acid and glycerol. And the important elements that make up the lipids are carbon, hydrogen, and oxygen. Okay? But for carbon, hydrogen, and oxygen, you'll have to remember that less oxygen, meaning less ratio of oxygen is present in a lipid. For example, if agar carbon in a lipid is C57, any 57 molecules of carbon, hydrogen, let's say it's 55. I'm not sure कि मैं बना क्या रही हूँ, but I'm just trying to explain one difference. And oxygen should be only 6. ठीक है? So among carbon and hydrogen, the least ratio you'll find is of oxygen in a lipid. ठीक है? So this is why we say that less oxygen is present in a lipid. Then it's a storage and it's also a structural molecule. And lipids are mainly responsible to provide mechanical protection for organs, because you see that, for example, if you have ever seen a kidney, then you can find kidney deeply embedded into a layer of lipid. So basically, lipids are a sort of mechanical protection for our organs, and they're large macromolecules. Then the examples of lipids that we'll have to study are triglycerides and phospholipids. We have already covered phospholipids in cell membrane. We'll study about triglycerides in this chapter. And these are like the examples of lipids. Okay? And why do we need lipids? So they're necessary for growth. And by nature, they're water insoluble. They always make up a layer in form of emulsification. And more hydrogen is present and less oxygen is present. So you try to remember these points. These are very important points, and they should be a part of your script as well. Because if, for example, someone asks you that what is the difference between lipid, protein, and carbohydrates when they're made up of the same elements, like they're made up of carbon, hydrogen, and oxygen. So how are they different? So they're different in terms of ratios of different elements. For example, in lipids, oxygen is less, but hydrogen is more. So this can be an inverse in some other, this can be an inverse scenario in some other biological molecules. Then they're hydrophobic. Hydro means water, phobic means fearing. So they are water fearing, and they're non-polar. Then they have low density. So why so try to understand this fact that you see that animals that live into the water, they can float easily, right? They do not just sink inside the water, but rather they can come above the surface of water. And why is that so? Because they are the lipid has a nature which is less dense. If lipids were denser than water, so all the animals that live in water, they would have sink. Okay? And this is also an organic molecule, and lipids have more energy. Uske baad, they act like insulating layer beneath our skin, and they form covalent and ester bonds. These are like small pointers that will help you understand the concept of lipids, and they can be asked to you in paper 1. And sometimes they also ask you about these pointers in paper 2, where you'll just have to write a brief introduction about lipids. Okay, so for 4 marks, for example, if you are given a question of 4 marks, then you'll have to give 4 solid pointers. For example, if you have to introduce lipids, you can say that they are made up of fatty acid and glycerol. You can say that in lipids, less oxygen is present, but more hydrogen is present. And then you can give examples of lipids, which are triglycerides and phospholipids. You can mention the fact that their water is insoluble, they are hydrophobic in nature, and they're non-polar. So these are the sort of introductions that are required in the scripts. Okay? So these are all the keywords, and all of them are equally important. Achha. So now let's move on. Anji. So this is a part which usually you'll do in your labs, but I've just written it. It's not the part of paper 1 or paper 2. This is a part of your paper 3s, and you'll do it in your colleges or in your labs. But let's just understand this fact that if you are given a test, if you have to do a test for lipids, then what you need to do is you need to take 2 centimeter cube of sample. Obviously, the sample is of lipids, right? Because you're doing the test for lipids. So 2 centimeter cube of lipid sample, and you'll have to mix it up with 2 centimeter cube of ethanol. Ethanol is an alcohol, and then you'll find a clear solution. And when you'll add few drops of water, you'll see a white emulsification being formed on the top of the surface of the test tube. Okay, so emulsion or emulsification is it's the final results of lipid test. Okay. So you take 2 centimeter cube of sample, you take 2 centimeter cube of ethanol, you add few drops of water, and you'll find a white emulsification, white emulsion layer on the top of the surface. Okay? Achha ji. So now let's move towards the important part. I just mentioned K formation wala process is known as, is it hydrolysis or is it condensation? Let's see who was active.
Miss, condensation.
Condensation. Excellent. So when we have to synthesize something, that process is known as condensation. Excellent, Arif. Okay. So when we have to make something, we condense it. And how are we supposed to condense it? We have to remove the molecule of water. Acha. So now let's see. Acha. Before moving on, let's just understand this word triglyceride. Tri means 3. Okay? Tri means 3. And glyceride over here refers to fatty acid chain. So triglyceride literally means that there are 3 chains of fatty acid. Okay? So as we have understood the fact that lipids are made up of glycerol and fatty acid chains. So if we're talking about triglycerides, then it should have 3 fatty acid chains. Like in phospholipids, when we were doing phospholipid bilayer, we talked about 2 fatty acid layers, right? But now we're talking about triglycerides. So now we are going to talk about 3 fatty acid chains. But now, in biological molecules, you'll have to remember their condensation as well, because sometimes they do ask this question. I remember that when I was doing my paper, I was not doing my paper of CI, rather I was attempting the paper of my midterm in my college. So there was this question that draw the formation of protein, and I was so confused because I never learned it too. And nobody told me that it could be a part of your paper. So we need to remember the formations as well. They do ask you to make this up in your papers. Okay? So for glycerol, you need to remember that glycerol is C3H8O3. Glycerol is C3H8O3. And you can see that you can see that there is less oxygen and more hydrogen, like it was mentioned in the previous slide as well. So glycerol is C3H8O3. So this is your first carbon, this is your second carbon, this is your third carbon, and you can see that we have 8 hydrogens around 3 carbons. One hydrogen, second hydrogen, third hydrogen, fourth hydrogen, fifth, sixth, seventh, and eighth. So we have 8 hydrogens and 3 oxygens again. Okay, so this is how you need to remember that C3H8O3 is constructed in this manner. Now we have fatty acid chain. So for fatty acid chain, you need to remember that you have a carbonyl group. You'll study about carbonyl groups when you'll study organic chemistry in AS. But for now, just understand the fact that this is basically a carboxylic acid. Okay? Carboxylic acid का functional group लगा हुआ है, which has a carbonyl and an hydroxide with it. So C double bond O and OH, this is carboxylic acid, and attached with it is R group. R group can be anything. It could be CH3, it could be like any length of carbon hydrogen. ठीक है? So जितने भी अगर आपने बहुत सारे carbon and hydrogen लगाने हैं, so it's better to write R instead of making the entire chain, because पेपर में इतनी जगह नहीं होती that you'll write so many carbons and then you'll attach hydrogen. जगह कम होती है, इसलिए we write R. So R represents any length of carbon and hydrogen. ठीक है? You can also write this in your notes if you're taking, that R is any length of carbon hydrogen. Or I should also write it. I just don't trust when I instruct students to write something, they just don't write it and then they make mistakes in their script. Any length of carbon hydrogen is known as R. And why do we write R? Because paper में जगह नहीं होती. So don't write paper में जगह नहीं होती, just write R. Any length of carbon and hydrogen. ठीक है? अच्छा जी. So now moving towards the fatty acid chain. So we have 3 fatty acid chains because we're talking about triglycerides. So यहाँ पर हमने लगाया carboxylic का group COOH and R2. फिर दोबारा हमने carboxylic लगाया and then R3. So this is R1, this is R2, this is R3. All 3 of them are any length of carbon and hydrogen, and these are carboxylic के groups COOH. ठीक है? Acha, now that we have to condense in order to make a triglyceride, what are we supposed to do? I'm changing the color of my pen to show you something, that we are now going to remove the molecule of water. So this is OH, right? And we have to remove one more hydrogen. इन्होंने उल्टा लगाया है, it should have been like this, HO. ठीक है? ताकि we can remove hydrogen from here. And this is how we'll remove water ka molecule. We'll again remove water ka molecule from the third one. Wait, let me write it this way. HO. Okay. So now we are again removing water ka molecule. So two water ke molecules यहाँ से remove, and then third one यहाँ से remove कर लिया. Again, I have to write this like HO. And this is how we removed 3 molecules of water. 1, 2, and 3. Just wait a second. Let me draw it and then you can see. Okay. So अब यहाँ पे क्या बचा? यहाँ पर बचा COC. O किसका? यहाँ पे O जो बचे थे ऑक्सीजन के, तो यहाँ पर it became COC. So now this bond COC is now known as ester का bond. ठीक है? So COC जब हमने H2O यहाँ से remove करा, we got 3 COC के bonds, and COC के bonds are known as ester bonds. And this is how you made triglyceride, by removing 3 molecules of water. अब पूछें.
मिस ये जो आपने एचओ इसको चेंज करें तो ऑर्डर ये लाजमी करना है या फिर हम किसी
नहीं
भी
नहीं,
ऑर्डर में लिख सकते हैं?
मैंने चेंज इसलिए करा बिकॉज मुझे शो करना था ना, वरना आप लोग कहते हैं कि मैंने ऑक्सीजन रिमूव कर दिया. मैंने इसलिए चेंज करा इन ऑर्डर टू शो कि मैंने H2O रिमूव करा है, हाइड्रो ऑक्सीजन नहीं रिमूव करा. ठीक है?
ठीक है.
कोई और क्वेश्चन, और शैल वी मूव ऑन? समझ आ गई कैसे बनाया ट्राइग्लिसराइड? ऑलराइट, सो नाउ लेट्स मूव ऑन. Acha ji, now we are proceeding towards carbohydrates. Carbohydrates, very simple to understand. Acha, after the namaz break, I'll ask few questions from lipids. So I'll check कि किसने कितना सुना है. ठीक है? So now coming towards carbohydrates, they are relatively simpler to understand. इतना मुश्किल नहीं होता. But अब थोड़ी सी slightly, obviously you are now in A levels. So there are going to be more technical things that we need to understand. Starting off with monosaccharide, we all know mono means one, di means two, and poly means many. Okay, so mono means one. And now if you're talking about monosaccharide, saccharide means a sugar unit. So we are talking about one sugar unit. And we need to remember the single unit of monosaccharide. So this is like the journal formula of monosaccharides. And this usually comes in your paper one, where they ask you the journal formula of monosaccharide. So it is C. Something happened to my screen. Wait. All right. So it's CH2 in subscript and O. Or उसके साथ we have N. So why do we have N? N could be like any number. For example, अगर यहाँ पे मैंने लिखा है 3, तो यहाँ पे वो 3 2 6 हो जाएगा, और कार्बन एंड ऑक्सीजन वो 3 के उस पे डिपेंड कर रहे होंगे. तो N could be like any number. So this is like the journal formula of monosaccharide, CH2ON. Then you have to remember that monosaccharides are small, soluble, and sweet. They have a crystal-like appearance. They are polar in nature and they are reducing in nature. Okay? Reducing in nature means that they can give results in Benedict test. Okay? Benedict test is done for monosaccharides and they can give a positive color change. Like they can give you the color change of red and orange. Red or orange rather, because they're reducing in nature. Reducing in nature means that they can give a positive result in Benedict. Okay? And the examples of monosaccharides are glucose, fructose, and galactose. We're moving towards disaccharide. You need to remember that disaccharides are also sweet, they're soluble, they're crystalline, but some of them are reducing and some of them are not reducing. So reducing means that they can give a positive test in Benedict, while non-reducing means that they cannot give a positive test in Benedict. Acha. So this is very important. You have to remember their formations, that glucose and glucose, when two glucoses combine, they form maltose. When glucose and galactose combine, they form lactose. And both of them, maltose and lactose, both of them are reducing in nature. But now moving towards the non-reducing sugar, which is sucrose. So when a glucose molecule and a fructose molecule, they combine, they form sucrose. And sucrose is non-reducing in nature. Okay? So we have a separate test for non-reducing sugars, and we have this test of Benedict for reducing sugars. We'll do the test of non-reducing sugars as well. But as for now, just remember that which two monosaccharides combine to form which of them, because this is a component of paper one once again. Okay? Now moving towards polysaccharides. So polysaccharides, all of the polysaccharides are non-reducing sugars. And they're not sweet, they're insoluble, they're not crystalline, and the example of polysaccharides are starch and glycogen. Okay? So far, have we understood everything? कोई मसला तो नहीं है?